Have you ever wondered why our hair is either curly or straight, and our skin is either white or brown? Well, you've come to the right place. That's what we're going to talk about today. Come and dive in with us. First, we need to talk about DNA and RNA. DNA, or deoxyribonucleic acid, is a molecule that contains the biological instructions that make each species unique. Also, the primary genetic material contained in our cells and practically other living organisms. The RNA, also known as ribonucleic acid, carries out the DNA-encoded instructions. Its fundamental role is to translate information into proteins. The transfer of genetic information states that the sequence involved in the expression of heredity characteristics is that DNA forms RNA, RNA forms protein, and later forms an organism or any living thing. Then, how are proteins produced? Protein synthesis is a process of making proteins. It also represents the major route of disposal of amino acids.
Now, what is the relation of DNA and RNA in protein synthesis? During protein synthesis, it occurs in two steps, namely transcription and translation. The first step is transcription. It is the process where a portion of DNA is unzipped, and the mRNA or messenger RNA is made of DNA template. Nucleotides of RNA will match with the first strand of DNA and make RNA. RNA polymerase is in charge of unzipping the DNA and puts RNA nucleotides into place. We must remember that in DNA, adenine binds to thymine, and cytosine binds to guanine. Two strands to know are the template or antisense strand, the strand where the RNA polymerase is acting on, and the non-template or sense strand, the strand that is not used. The mRNA strand is then capped at its 3' end by the poly A tail that contributes to the life of transcripts. The 5' end is then capped to protect the new strand from enzymes that can destroy it, and the pre-mRNA is now produced. Before the pre-mRNA enters the cytoplasm, it must be edited first. Editing is where nucleotides that can't be coded, called introns, are edited out by an enzyme, while the codons that are left, or exons, are spliced together by the spliceosomes. After the transcription process, how does translation take place?
After transcription, the next step is translation. It is the process where the mRNA is attached in the ribosome and is approached by the tRNA or transfer RNA that carries amino acids where the bases of the mRNA strand are read in triplets that is also known as codons. And the tRNA deposits an amino acid into a growing chain of amino acids called the polypeptide chain. We must also keep in mind that in RNA, adenine binds to uracil and cytosine binds to guanine. The tRNA with its anticodon UAC is attached to AUG that serves as the start codon that codes for an amino acid called methionine. Well, that was quite the journey, wasn't it?
You now have a concept on how the information stored in DNA is used to make protein.
That's all for today. We hope you learned something and had some fun too.
Thanks for watching. Bye-bye.